Hi, I'm Maria Littmann. I'm a postdoc at the Technical University of Munich, and this is our poster on using protein embeddings to predict binding residues for various ligand classes. Here we want to propose the method BindEmbed21, which predicts binding for 3 different ligand classes, namely metal ions, nucleic acid, and small molecules. BindEmbed21 consists of 2 components. The machine learning component utilizes the pre-trained transformer model PROT-T5. This model was developed and pre-trained by Ahmed El-Naga and Michael Heinzinger, and the model can be used to extract per residue representations, so-called embeddings, for a protein sequence. Those embeddings, with 1024 dimensions, can be used as an input, or are used as an input to our machine learning model, which is a 2-layer CNN that then predicts whether a residue is binding to metal ions, small molecules, or nucleic acids. The second component of BindEmbed21 is using homology-based inference to infer binding residues from a sequence-similar protein with annotations to our query protein without known annotations. BindEmbed21 combines these 2 components so that, if possible, we always apply homology-based inference, and if we can't find a sequence-similar protein with annotations, then we fall back to the machine learning method to make a de novo prediction. This combination in the end reaches, for example, an F1 score of 48 plus minus 3%, and this is 5 percentage points higher than just using the machine learning method, which is indicated here with this lighter colored bar. And in fact, if you look at the full performance assessment, you can see that using this combination of machine learning and homology-based inference always outperforms just using the machine learning method, except for the precision here for nucleic acids. BindEmbed21DL is solely based on single sequences, and we compare it to BindEmbed17, which uses evolutionary information encoded in MSAs. And as you can see, for all 4 performance metrics, our method BindEmbed21DL outperforms BindEmbed17. So you can see that even though we are only using single sequences, we can outperform machine learning. We can outperform machine learning methods based on evolutionary information in MSAs. Also, BindEmbed21DL predicts 3 different ligand classes, and many other methods actually specialize on 1 ligand class or a set of ligands. For example, PRONA2020 predicts binding to proteins, DNA, and RNA. And we compared its DNA and RNA prediction to the DNA and RNA binding predictions of BindEmbed21DL. And you can see that BindEmbed21DL is at least competitive to this specialist. So we achieve a higher precision but a lower recall, and this leads in the end to a similar F1 score and a slightly higher MCC. So BindEmbed21DL does not clearly outperform PRONA2020, but you also have to keep in mind that it is not specifically trained for this task, and that BindEmbed21DL provides much more information about the binding residues, because it also predicts binding to metal ions and small molecules. And also PRONA2020, similar to BindPredictML17, is based on MSAs, so the method is much slower, and you also see another indication that BindEmbed21DL can in fact outperform evolutionary information-based methods using single sequences. In fact, we can also alter the performance of BindEmbed21DL by altering the output probability that we consider to define binding. So usually everything below a probability of 0.5 is considered non-binding, and everything above is considered binding. And if you decrease this probability, the recall increases, so more binding residues are predicted. And if you increase the probability, the precision increases. And for example, the output probability of 0.95, BindPredictML17 reaches a precision of 73%. Since BindEmbed21DL is very fast, we can also easily apply it to full proteomes, which is what we did, for example, for the human proteome. For the human proteome, only 15% of all proteins have any binding annotation, and this even means that we are considering structures with a rather low resolution or NMR structures. We can extend this fraction of proteins with binding annotations to around 50% if we also take homology-based inference into account. But this also means that we are applying a rather high e-value. If we apply a more realistic and stricter e-value cutoff, then we are at something around 40%. Of proteins with binding annotations. If we apply BindEmbed21DL to predict binding residues, we actually get predictions for 92% of the human proteome, and 42% of the proteins did not have any binding annotation before. So we only have the binding predictions for those. And in fact, of those 42% new predictions, or proteins with new predictions, 30% of the proteins have at least 1 binding residue predicted with a reliability or probability above 0.95. So these are very highly reliable predictions. And also based on our predictions, we can estimate that 1% of all residues in human proteins is binding to metal ions, 2% are binding to nucleic acids, and 3% are in fact binding to small molecules. So in conclusion, with BindEmbed 21DL, we have a method that uses transfer learning solely based on embeddings to predict binding residues, and this actually outperforms methods that use MSAs as an input. We can further combine this machine learning method with homology-based inference to boost performance to a final F1 score of 48 plus minus 3% and an MCC of 0.46 plus minus 0.04 to predict binding to metal ions, nucleic acids, and small molecules. And given our analysis for the full human proteome, we would propose that 6% of all residues in human proteins are involved in binding in some way. BindEmbed 21DL is also publicly available. The source code is available via the GitHub repository BindPredict, and then BindEmbed 21DL as well as homology-based inference and the combination of both methods. BindEmbed 21 is integrated into the bioembeddings pipeline, which allows easy use of the method and its components because bioembeddings provides examples and also configuration files that can easily be executed to run predictions. Last but not least, I would like to thank ISMB for giving me the opportunity to present my work. I would like to thank my co-authors for working with me on this project, and I would like to thank all of you for your interest in our research. If you're interested in learning more about BindEmbed 21, there is also a talk in the Function Cozy from Monday, July 11th, where we explain more about the method and provide more results. Thank you all for listening.